Okay, so we talked about in the last video the importance of collecting raw data with 5 increments. So in that photosynthesis simulation, we did 5 different light intensities, and we saw the effect of the rate of photosynthesis based on those 5 increments for our IV, right, for our independent variable. Sometimes your independent variable can just be 2 things. So in this video, we're going to talk about how do you collect meaningful data, meaningful raw data, when you're only looking at two independent variables, like 2 increments. So I'm going to scroll down just so we can draw a little bit. So sometimes you might have a situation where you're only looking at A or you're looking at B. So this could be boys or girls, or it could be your right hand versus your left hand. Like there's nothing really in between the twos. You can't have increments along the way. So a very common one is like maybe at location A, you're looking at a certain species and you're looking at the same species at location B. And maybe you want to know what's the population difference in these 2 areas. And maybe location A is like near a river and location B is like on top of a mountain or something. So those are very 2 different environments. So maybe that would affect the distribution or the population sizes of that species. Or it could be biodiversity or whatever. It could be lots of different things you're looking at. So when you are doing this, so location A and location B, you have to make 15 different measurements. You have to collect 15 pieces of data all the way down to 15. And eventually, at the end, you're going to be calculating the average, or the mean, as we scientists call it. And this is what you're going to compare. You're going to be comparing the 2 means, in this case, the population of a certain species at location A and the population of a certain species at location B, or of the same species at location B. So that's what this means. How do we collect raw data when we're comparing 2 means? So we're going to practice this using a pretty useful website called Human Benchmark. Human Benchmark has a lot of different tools on it that might be useful or maybe a resource for you to do some sort of investigation. These are looking at cognition and how neuroscience works. So we're talking about short-term memory here. Maybe you want to look at the human perception and hearing. So how does hearing loss, or how does age, affect hearing loss by measuring the person's ability to hear certain frequencies? This one's kind of cool. So there's a bunch of different ones there. We are going to use reaction time. So for this experiment, I am just going to see, is there a difference between the reaction time of my left hand clicking when this changes from red to green versus the reaction time of my right hand? Maybe not the best experiment. If you decided to do this as your IA, I probably would talk you out of it. I would ask for something a little bit more sophisticated, but just a way for you to see how this works. So using this, I'll demonstrate it now. So when you click on this box, the screen will turn red. And as soon as you see it turn green, you click again and you record that value. It'll give you your reaction time in milliseconds. All right, I'm waiting for it to turn green. Okay, 393. So I have created like a little Google sheet here. This is my left hand. So I would enter 393, 393. And then I would do it again. So I click this, I wait for it to turn green. I'm getting faster. 277. So then I write 277 and so on. And I would do this for 15 trials. And then eventually, and if you want, you can do this. I'm not asking you to do any kind of data, process data. So we're not going to do any data processing yet, but you're welcome to if you want. So collect those 15 measurements for your left hand collect those 50 measurements for your right hand. You're welcome to be creative and think of something different. Maybe right eye closed, left eye closed. Maybe maybe you're trying to multitask by, I don't know, reciting the ABCs as you're waiting for the reaction time. So there's a bunch of different ways you can be creative here. Yeah, so that's how we collect data with comparing two different situations by looking at their means.